Okay, this will be our notes over photosynthesis, and I'm going to apologize now because I'm losing my voice, so this does not sound that wonderful. Anyway, photosynthesis is when plants or autotrophs convert sunlight into cellular energy. And here's an overview of what goes on. We have energy from the sun coming in as light, striking the chloroplast, and they then make organic molecules and release oxygen that is used here for the mitochondria, for the sites of cellular respiration. ATP is made, gives off carbon dioxide and water, which is then again used by the autotroph. And we lose some energy here as heat. Okay, to talk about photosynthesis, we have to talk a little bit about light. So this is our visible light spectrum right here. Sunlight transfers radiant energy through discrete particles called photons or waves. There's a wave-particle duality of light, where some argue that a light wave is just simply a wave, and some people argue that there's little packets of material called photons that are light. Now, the wavelength from crest to crest or trough to trough determines the color that the light will be. So as you can see here, the lower the wavelength, the higher the frequency, or the more energy it has. Okay, and then the longer the wavelength, it goes here to the red side. Photosynthesis takes place in the chloroplast, which is an organelle in autotrophic organisms. It's composed of a double outer membrane. The inner folded membranes are called the thylakoids. This is where, when we talk about the light phases that we talk about next, that's where those occur. And a stack of thylakoids is called a grana. And then the fluid interior of the chloroplast is called the stroma, whereas in the mitochondria it's called the matrix. Here it's called the stroma, and that's where the Calvin cycle takes place. And just a side note, spectrophotometry, it's an equipment that shines a light through a solution that has a pigment, and then it will measure the amount of light that's absorbed or transmitted at a specific wavelength. And as we would guess, most plants that we know of are mostly green in their leaves, so that's where most of the chloroplast can be found. And it's going to be found in the palisade layer and the spongy mesophyll, right here in this section, and the spongy mesophyll is down here. Okay? And here's our reaction that is actually what's photosynthesis. We have 6 carbon dioxides, 6 carbon dioxides and 6 waters, with that light energy coming in, obviously. Light energy coming in gives us glucose and 6 oxygen. Okay, there's 2 different parts to photosynthesis. There's the light reactions and what's called the dark reactions. So we're going to talk about the light reactions first. This is what happens first. Chlorophyll A is a reaction center, and chlorophyll is just a pigment, okay? When you see that a leaf is green, you are not actually, the leaf isn't actually itself green. It has the pigments in it that are going to absorb the blue violets and the reds and then reflect the green. So your eye actually sees that green wavelength. So you're actually seeing the reflection of green, if that makes any sense. An electron is going to get excited when the light hits it, and it jumps out of orbit into a higher energy level. This is called photoexcitation. It's being excited by light. Then a primary electron acceptor will grab that electron and send it down an electron transport chain, similar to the electron transport chain we saw in cellular respiration. And here, this is what I was talking about, the light reactions. And in reality, we have photosystem 1 and photosystem 2. They were named in that order. If you read in chapter 10, you would have read this. They were named in the order that they were discovered but this one actually occurs first and that one occurs second. So P680 is excited first here in photosystem 2. Then that primary acceptor sends the electrons down electron transport chain, and there P700, same thing happens here. It's excited and it loses, or an electron jumps out of its orbit. And these light reactions are going to produce ATP, and that should say NADPH. NADH is in cellular respiration. NADPH is in photosynthesis. Sorry about that error. And then just talking a little bit more in detail about how those light reactions occur. A photon of light strikes the reaction center. That electron is captured by the primary electron acceptor, and then water is split to fill the hole that's left by the loss of the electron. So then you have this hydrogen ions there that are lost as well. So then the electron from P680 falls down the electron transport chain and fills the hole in P700. And then the electron transport chain produces ATP through photophosphorylation. It's not substrate level phosphorylation like we saw in cellular respiration. This is photophosphorylation because it is caused by the photons striking the P680. The photon also will excite an electron in P700. That electron is captured by the primary electron acceptor and sent down the electron transport chain. And again, this down here should say NADPH. So sorry about that again. And here's a picture of the light reactions. So you have this photon of light hitting P680, the breaking down of water, these two electrons that are excited. This electron acceptor is going to grab it, carry it down this electron transport chain. We also have light hitting P700 combined with this, so we have again the excitation of the electrons. And here is where we'll lose the two electrons, and NADP, there it is, becomes NADPH. You can check those out through my website. Okay, and then the dark reactions. This is when the carbon, the CO2, is actually fixated inside the chloroplast from the atmosphere. These reactions are driven by ATP and NADPH, and it's called the Calvin-Benson cycle, or sometimes it's just called the Calvin cycle. When the CO2 is fixed, it's going to combine with something called 5-carbon ribulose biphosphate, which is shortened into RUBP. And the enzyme that catalyzes this is called rubisco, and this is believed to be the most abundant protein on the whole planet. And these reactions occur in the stroma, so that fluid-like material inside the chloroplast, similar to the matrix of the mitochondria. And here's a picture of the Calvin cycle. So as you can see here, these are the light reactions, and this is the Calvin cycle. And you've got the ADP and the NADPH and the ATP and NADPH. Sorry, that was NADP up there. Okay, so here you have the carbon being fixated using rubisco. It combines with this RUBP, and it creates this 6-carbon intermediate that is so unstable it immediately splits into 2 3-carbons, the 3-phosphoglycerate. Okay, and it shows 6 here because this is happening 3 times. All right, so when it splits, then now we have 6 of those things. So we're going to 6 ATPs are going to be reduced to ADPs. So these phosphates are added on here, so it becomes 1,3-biphosphoglycerate. And then again, we have the same thing happening here, NADPH becoming NADP, losing a phosphate. So now we have glyceraldehyde 3-phosphate, which is G3P. Okay, this is going to be, do you see there's 6 of them here? How many are coming out of the cycle? Just 1. Because the other 5 have to go back this way, and they're going to be rearranged and formed using ATP again to make this RUBP, to recycle it so that it can be combined again with the fixated carbon. And down here you'll see that the whole point is we're making glucose, right? So how many of these would you need? Well, you'd need that to happen twice to get a 6-carbon sugar. And this is saying it all again without looking at it, so we'll just go ahead and I'll walk you through it again so you can get it written down. Like I said, that 6-carbon molecule is so unstable, it splits into 2 3-carbon molecules. Phosphates from ATP are added to the 3-carbon compounds, and NADPH oxidizes the compounds. G3P is that 3-carbon sugar that's coming off that down the bottom of that last slide. So you have 3 molecules of CO2 entering the Calvin cycle, resulting in 6 G3Ps, but only one of them is released. And like I said, those remaining 5 will gain a phosphate from ATP and rearrange their atoms and eventually become the RUBP again. So a total for the Calvin cycle of 9 ATPs is used and 6 NADPHs are used. And this is just another look at it again. And this is where the 6 are used here, 3, so we have 9 ATPs used, and then the 6 NADPHs. Now, most plants are C3 plants, meaning the first molecule of the Calvin cycle is a 3-carbon compound, just like we just saw. But sometimes you have levels of CO2 declining because stomata close, and so then we might have something called photorespiration occur. And this is when the plant will fixate oxygen rather than carbon dioxide. This process produces no sugars or ATP and decreases the efficiency of photosynthesis. To avoid the cost of photorespiration, some plants produce an intermediary molecule that allows the dark reactions to occur with the stomata closed or at night. The 2 examples that we have here are the C4 plants, and these produce phosphoenol pyruvate. It's shortened to PEP. That's much easier to say. And that's a 4-carbon compound. That's why they're called C4 plants that can release CO2 into bundle sheath cells so carbon dioxide levels remain high enough for carbon fixation to occur. And the examples in real life of this would be corn and sugarcane. And then the other type we have are called CAM plants. They take up carbon dioxide at night when the stomata are open, and then they store that CO2 into compounds that will later during the day release the carbon dioxide so that when there's enough ATP and NADPH with the sunlight, it can produce the sugar. And the example here is the cactus. And that is our notes for today. Hope you enjoyed it. Bye-bye.